Western blotting is the detection and visualization of a specific protein with the use of antibodies. It is known as the most common technique for protein detection. In order to perform Western blotting, a sample of proteins must be first transferred to a membrane. Two techniques used to do this are microfiltration and electrophoretic transfer. Microfiltration is when proteins in solution are transferred to a membrane without first resolving the proteins. This is a suitable method for when a large number of sample needs to be screened or when a new assay needs to be tested. More commonly used is electrophoretic transfer, which is a precise and quick way to replicate protein bands that have been resolved on a gel. To begin, sodium dodecyl sulfate polyacrylamide gel electrophoresis, or SDS-PAGE, needs to be performed to separate the proteins in the sample based on size. Proteins have regions of positive and negative charge towards the outside and hydrophobic regions in the center. In order to run the proteins in the gel based on size alone, they need to be denatured and they need to have a uniform negative charge. Adding beta-mercaptoethanol, SDS, and then boiling the sample achieves this goal. The protein is now denatured with a net negative charge. The protein samples are added into the wells of the gel and the gel is run. The gel has an anode and a cathode, so when an electrical current is applied across the gel, the proteins migrate down the gel towards the positive end based on size and form bands. Smaller protein samples travel farther than larger ones. Next, the proteins need to be transferred from the gel to a membrane for detection, which involves something called a gel sandwich. No, not this kind of sandwich. This kind. The membrane the proteins are transferred onto is called a nitrocellulose membrane, which has a high affinity for proteins. Protein binding with nitrocellulose is instantaneous, irreversible, and quantitative. It was the first type of membrane to be used for Western blotting, and it is still popular. Another type of membrane that is sometimes used is called polyvinylidene difluoride, or PVDF. It has better protein binding affinity and has many other uses besides Western blot. It is also reusable. There are 3 types of apparatuses used to facilitate transfer, but they all use the same elements in their gel sandwich. The 3 main kinds of electrophoretic transfer systems are dry transfer, semi-dry transfer, and wet transfer. Wet transfer uses a tank of transfer buffer to submerge the gel sandwich. This method is the most efficient and has the most flexible settings. It also works for proteins of all sizes. Semi-dry transfer uses a sandwich soaked in transfer buffer. This method is more convenient but lower quality. It also might have problems transferring very large or very small proteins. Dry transfer uses pre-assembled stacks of buffer matrix in its gel sandwich. This method is very fast and produces good quality results. Dry transfer might take 10 minutes, while semi-dry takes up to 3 hours, and wet can take longer than 4 hours. Dry transfer using the iBlot is favorable because it is fast, simple, and reliable, while the other techniques can take much longer and require more work. The copper plates on the iBlot machine are used as electrodes. This gel sandwich is placed into the machine and the lid is closed. Now for a close-up of what's happening inside the machine. When the iBlot is turned on, the copper electrodes send electrical current through the gel sandwich, and the proteins, which are still negatively charged, transfer to the nitrocellulose membrane. Once the proteins are transferred from the gel to the membrane, a reversible Ponceau stain can be performed for visualization. All the bands from the gel have been transferred. Here are the proteins on the nitrocellulose membrane. In this example, purple represents the protein of interest, which will be detected using Western blot. The membrane is first blocked with a protein that will not interact with the antibodies used to detect the protein of interest. BSA and casein are 2 kinds of blocking buffers commonly used. They prevent the antibody from binding nonspecifically to the nitrocellulose membrane so that it binds only to the protein of interest instead. The blotting solution used in this case is casein, which is inexpensive and widely available because it is found in non-fat milk. Casein is added to the membrane. Now, add the antibodies to probe the sample. But first, how do antibodies work?
Antibodies are proteins that bind specifically to a certain protein. The protein an antibody binds to is known as an antigen. Each antibody contains a variable region, which binds to foreign antigens in the body, and a constant region, which is the same on all antigens found within a species. The antigen binding site in the variable region has a particular shape that binds to a specific antigen. The epitope is the region on the antigen that the antibody recognizes. Only antigens with an epitope of the right shape will bind to the antibody.
The blot is first probed with the primary antibody, which binds to the protein of interest. Once any unbound primary antibody is washed off with buffer, the Western blot is probed with the secondary antibody. If the first antibody was made in a mouse, the second might be made in a goat. This secondary antibody will attack any antibody it recognizes as foreign, which in this case is a mouse antibody. The secondary antibody recognizes and binds to the constant region of the primary antibody, which is the same for all mouse antibodies.
Now that the protein of interest has been probed, it needs to be visualized or detected. 3 types of detection commonly used are colorimetric, fluorescence, and chemiluminescence. In colorimetric detection, the antibody is conjugated to an enzyme like horseradish peroxidase. When the blot is incubated in a colorimetric substrate, such as chloronaphthol, bound horseradish peroxidase cleaves the chloronaphthol, producing a purple precipitate. Other enzymes and substrates could be used as well. It is not always necessary to use a secondary antibody in Western blot, but it is helpful. Since multiple secondary antibodies can bind to each primary antibody, more chloronaphthol is cleaved and more purple precipitate is produced, therefore amplifying the signal. Fluorescence is another technique used. Fluorescence detection uses an antibody tagged with a fluorophore, which emits fluorescent signal when it is excited by a light source, which is captured by a camera. In chemiluminescence, the reaction of a substrate binding with the antibody produces light as the signal, which can be captured using X-ray film. Now for the end result. A Ponceau stain performed before the antibody probe shows bands representing all of the proteins present in the sample. Western blotting only shows bands that correspond to the specific protein of interest. A Ponceau stain might look something like this. Now the corresponding Western blot. Bands representing the protein of interest can be seen. Smearing may appear due to partial degradation of the protein.